I'd like to welcome everybody to 599J today, and especially welcome Dr. Charles Cow to Seattle, who will be our speaker today. Dr. Cow received his PhD at Jilin University in China, where he won two very special awards. He was first named the Outstanding Graduate Student, and then he also won their Excellent PhD Thesis Award. After he graduated from Jilin, he moved on to the Hebrew University in Jerusalem, and he conducted a two-year postdoc there with Professor Yuri Bannon. And that was under another nice fellowship award, the Golda Meir postdoctoral fellowship. After that, most recently, he moved to the States, where he's been working with Chad Merkin at Northwestern University. And they have a nanoscale science and engineering center that was one of the first bio-nanotechnology awards. And so he's done lots of exciting work there, as well as previous to that in Israel and China. Dr. Cow will be giving a talk entitled Nanoparticle-Based Detection, a New Frontier in Biodiagnostics. Okay. Welcome, Charles.
Thank you. It's really my pleasure and my honor to speak here. So many people. I'm really excited. In last 4 years, my work focused on the field of colloidal nanoparticles, which is a central part of nanotechnology. In this talk, I will tell a little bit about this content inside this, beside this title. I will introduce you a little bit broader perspective for nanotechnology. In Hebrew University, first I involved to develop a new synthetic strategy for making indium arsenide cultural nanoparticles with emit in near infrared, which is very important for telecom application and also for bio labeling. Using scanning tunneling microscope, we can identify S and P states in these nanoparticles. In addition, using scanning tunneling microscope, we can direct visualize this separate state. So after this work, I moved to States, work with Chad Merkin. So my work focused on using microparticles to do bio-labeling and bio-detection. So first, we discovered how to make triangular silver particles, and also using cultural strategy to stabilize silver spherical particles and use as bioprobes to do DNA detection. Finally, this is a very exciting part, using scanning Raman probe to detect DNA, RNA, and protein. And today I will focus on this part, but I briefly introduce you how to make triangular particles and a little bit about the electronic structure of semiconductor nanoparticles. So can you hear me, or I'm a speaker? Okay, so that's fine. So all my stuff is based on nanoparticles. To give you a feeling about nanoscale, I want to show you the size of things. A penny is 2 centimeters. It's easy to see. And the fillet is 1 millimeter. The diameter of a hair is 50 micrometer. Then a blood cell is 5 micrometer. Finally, here is 5 nanometer of carbon selenide nanoparticles. The typical size of colloidal nanoparticle located from 1 nanometer to a few hundred nanometer. So this is my first question is, why such small particles are important? So first, they have the size, shape, and the composition tunable properties. Here show example. cadmium selenide nanoparticle, the emission can be tuned from red to blue by just reducing their size due to quantum confinement effect. This is just a beautiful part of nanotechnology. One can use one materials, simply tuning their size. One can generate a series new fluorophores with color covered out visible spectrum regime. In addition, these particles are dispersible in solvent, so one can use this benefit to assemble them into larger structures to build function materials. So usually these colored nanoparticles are referred as a building block for nanotechnology. To synthesis of high quality nanoparticles and understanding of fundamental properties, pursuing applications are three big issues in this field. Today, my talk will focus on the three big issues which is related to my work. And in the first part, I was showing nanoparticle synthesis. Think about the time issue I want to start. I will skip this indium arsenide nanoparticle synthesis. I will start from an isotropic nanoparticles synthesis. And in the second part, I will show you atomic-like structures of semiconductor nanoparticles. And finally, I will focus on this using nanoparticles to do biodetection. So first, I start from an isotropic nanoparticles. Typically, it's easy to make spherical particles. For the particle with the other shape, it's hard to make. And I summarize the successful strategies for making an isotropic nanoparticles. First is using a membrane template synthesis. Charles Martin from University of Florida can make nanowires and nanotubes, and Mike Nathan used this strategy to make metallic buckles. And in the second category, one can use micelle template synthesis. One can make cubes, pyramids, and nanorods. Then the third one is physical and chemical deposition. Charles Lieber used this strategy to make nanowires. The third is kinetic control growth. One can make nanorods and nanocubes. But if you summarize this strategy, you will find it's mainly focused on the strategies to make nanowires and the nanorods. But how about the other shapes? It's hard to synthesize. Now I will show you how to make triangular silver particles. So in this flask, it contains a typical silver colloid, which is the size from 6 to 8 nanometers. And the shining with fluorescence light, it's just regular like here, this light. And after 3 days, what happened? Their optical properties gradually change. They absorb, actually they turn to blue. And what happened? So we go to the TEM. We found we get beautiful triangular particles. And each of those particles has a flat top and bottom. And because they're so flat, due to the van der Waals interaction, they tend to assemble to stack here. So you can see the stack. When you turn this, you can find it still is a nanoprism. Each of these triangular silver nanoprisms are beautiful single crystal. They have HCC structures. If you take careful look of this electron diffraction pattern, you will find has an additional side, which is 4 to 2, one third 4 to 2. Which is means this is the triangular particles has atomic flat top and surface. This is a typical phenomena from the thin prism has additional electronic diffraction dots beside the regular XCC structures. Then what is the mechanism? Why we get this triangular shape over than the other shape? So this is really hard for the mechanism. I use a lot of my time to pursue this mechanism. Right now I have a brief mechanism. The first, this is 6 to 8 nanometers spherical particles. On the light, they dissolve due to Oswald-Rappening. And they found some smaller nucleus, triangular nucleus, then the nucleus grows larger to final size of triangular particles. And this process are photochemical reaction. Turn off the light, nothing happens. And the size of starting silver nanoparticle is important. If this particle, if you use 20 nanometer, it doesn't work. You have to use smaller particles. And this phase selective growth. The top and bottom are 1-1-1 phase. So this gave us a triangular shape. And beside this, we found this is not only simple light excitation reaction. It has a surface plasma resonance associated process, which means if we use different wavelength excitation light, probably you will get different things of your product. What we get, can you imagine this? We found use different wavelength excitation, we got different size of nanoparticles. It's really beautiful. And here is our product. This is a use 540 nanometer excitation light. We get 30 nanometers, and use 650 nanometer. We get 120 nanometer size particles. And this kind of size control is brand new, and we are pursuing this mechanism. So basically, it's due to the dipole resonance excitation of its particles. When the dipole excitation is going to a red shadow of this excitation light, and they stop this growth. This is really fascinating. Then next things we have to understand the optical properties of each nanoprism. What is the meaning of these peaks? Here I will show you the experiment result compared with the spherical particles. You see, the nanoprisms has 3 peaks, and what is totally different with the starting point? What is the meaning of this small peak and this larger peak? We carry out a theoretical calculation, use discrete dipole approximation. So we found the first small peak is optical quadrupole resonance, and this peak is in-plane quadrupole resonance. The big peak is the in-plane dipole resonance. Because of the in-plane dipole resonance at 670 nanometers, so they should sketch light in right. So then if you combine with silver or gold particles with different sizes, you can build a beautiful scattering trains. So this is silver nanoprisms. And this is fundamentally important for, this is the first diamond for microparticle. You tune the size and shape and combinations, you can get different scattering light. Technologically important, you can use each of these particles as a biomarker. So say if you functionalize 6 different oligonucleotide probes, then you can simultaneously detect 6 probes in one solution. So this is I finish about particle synthesis. In the next part, I'm going to atomic-like structures of semiconductor nanoparticles. For semiconductor nanoparticles, this is back to 1980s from early theoretical work carried out by Louis Bruce. He predicted that in this semiconductor nanoparticles has separate electronic states with narrow transitions, and this state has S and P characters. And later, optical works prove that they do have separate energy levels and narrow transitions. However, the optical measurement could not directly give you the symmetry of this state. You don't know this exactly if it's S or P. Right now, I will tell you a technology which is using scanning tunneling microscope can provide you directly information about this identification. So what I do, I first attach indium arsenide quantum dots, we call them nanoparticles, on the surface of gold, and probe by this TEM tip from this measurement confirmation has two junctions. When you change this bias values to certain numbers, they have single electron tunneling occurs. For the negative bias, the tip actually pump electrons from the dots. So this provides information about the valence band of these quantum dots. For the positive bias, the tip inject electrons through the empty orbitals of these quantum dots, provide you information about conduction band. So today I will show you really simple things focus on this conduction band levels. Here's a typical scanning tunneling spectrum for the conduction band. You can find this basically has two groups. Inside group has a basically similar separation between the peak. If you carefully characterize the separation, you will find this intergroup separation is correspond to the single electron charging energy, and this is a larger separation is due to discrete quantum dots level spacing. Then you should think, if you carefully take off this energy lift by single electron charging energy, you will find the degeneracy of this quantum dots at first energy level is 2, and in the second level is 6. So if you remember Pauli principle, you will find this is S character. Then the second one should P. You have 6. So this S to P failing sequence exactly followed atomic like Aufbau principle. So now once you know this quantum dots are artificial atoms, you can think about how to use artificial atoms to build molecules, to build artificial polymers and liquid crystals and the crystals. So this is really fundamental for whole nanotechnology. So next I will show you using scanning tunneling microscope, we can directly visualize this SMP states. Here I will show you our result in indium arsenide and zinc selenide Cauchy nanoparticles. We simultaneously get 3 tunneling current image and topography image. For the first tunneling current image, we set this bias at the S orbital, and the second we set in middle of P, and the third one is end of P. And what is the meaning of the tunneling current image? Are they really image of these orbitals? For the S orbitals, easy. They are spheres, so we get spherical-like stuff. And what about this P? Why does it get these donor structures? And why they turn back when you raise the bias a little bit? So for this purpose, I carry out the calculation, use a particle in spherical box mode, and the radial part spacing used in these figures. And the results really surprised. Exactly reproduce our experiment data. So this is s orbital, okay? And for the p, we understand this because the mixture between the px and the py, we get this donut structure. And for the pz, they go back to spherical again because this kind of geometry. And so now I finish about the two part. The first part is nanoparticle synthesis. The second part is electronic structure of semiconductor nanoparticles. Now I will focus on this third one, using nanoparticles for biological detections. The biomolecules, here I will show you examples like DNA. For example, DNA, they do not provide stronger physical signals. So that means when you want to measure them, to detect them, you have to use labels. The current commercialized labels include isotopes and colorimetric detection and chemiluminescence and fluorescence detection. The fluorescence detection is the core technique for current applications. Today, I will show you a brand new technique which is built on nanoparticles, which has an advantage over this most popular technique, current technique, at simple selectivity, sensitivity, multiple labeling capabilities, and ratioing capabilities. The first, I will show you how to make nanoprobes. So this is a typical gold colloid capped by citric acid, and you put this particle react with cell function like oligonucleotide. Then you got the cell functionalized on the gold surface, and this oligos lay down on the surface due to the electrostatic interactions. And this kind of probe is poor functionalized. We call it poor function. It's not stable. If you want to make stable probes, so you have to add salt to wake this electrostatics. So you give us more room for the initial for the addition oligonucleotides going to form this heavily functionalized probes. So this kind of functionalized probes is our starting point for the result what I'm talking today. So before I joined working group, they developed a detection by following principles. The two types of gold particles with functionalized different oligonucleotide. So this is A, which is complementary with the target A prime, and the B is complementary to this B prime. So once you have the target, so this probes tend to aggregate. This aggregate process with significant color change from red to blue. The origin of this color change is due to the surface plasma band, this red shape and the damped. And using this technique, they developed really interesting things. It's called litmus paper for DNA. So you drop this solution onto a TLC plate without target. So this negative is red, but positive is blue. So this is a solution-phase detection. They also develop a technique allowing you to do chip-based detection, microarray-based detection. So this use silver staining associated, we call the schematic detection. This detection has really high sensitivity. And another one can use conductivity measurement, has really high target selectivity. So now I summarize the comparison between our technique with fluorophore dyes. You can find for the sensitivity and the selectivity, the Merkin technique can win. However, for these colors and the other three category, he couldn't win. So now I will tell you how can I win this category and still with the selectivity and sensitivity? Let's use Raman-based detection. So first, I functionalize a Raman dye labeled oligo probe on the gold particles. And this capture was attached on the surface of the chip with a desired target. So the particles will attach on the surface of the chip. In this case, usually we detect only very low target concentrations. So this means only few of gold particles on the chip surface is hard to detect them. However, if you do silver staining to grow silver particles around these gold particles, you will find the chip area with gold particles tend to black and give you really strong surface-enhanced Raman signal. To give you a feeling about what the surface-enhanced Raman scattering, I borrow a slide from Rick Van Dyke, who discovered this phenomena from Northwestern University. And I won't go through these two detailed things, but I want to show you two things. On a chip surface, on a surface with particles, close-packed particles, the Raman scattering can be scattering enhancement can be really high, up to 10 to 14 times enhancement are available. However, Rick's techniques basically on this electron chemical process substrate. So that means it's hard to control the quality of his techniques. It's hard to get reproducible results. Then I will show you how can I solve his problem and get for biodetection. Here is a typical chip after target and probe hybridization. You couldn't see anything. However, after silver staining, this process, the black spot come out. Then you measure the Raman spectrum from each of spot. You will find has very huge scattering signals come up, which is only come from the label, the CY3 molecules. It's not from oligonucleotides and other chemicals which are used for this detection. So that just gives us advantage. So we can use this Raman spectrum of this dye, this CY3 dye, as a fingerprint to monitor this specific target, experience of specific target. So say if you have many type of probes, so you labeled with different Raman dyes, then you use the Raman signal from each Raman dyes as the encode information to probe bunch of different targets. So when you scan, when you scan through this probe, this chip, so you can get a very nice intensity profile with distance. So this kind of intensity profile was got by our first generation of Raman microarray scanner. So by this scanner, we can simultaneously get Raman spectrum and this intensity profile. So here I will show you when you move this probe through the chip, you will find only on the black area you have Raman signal. And from dot to dot, we have very good reproducible. And after this presentation, this movie, I will show you the result about the detection limit. And this is typical detection carried out at 20 femtomolar. Femtomolar means 10 to negative 15 molar, really low. And we still can get very good signal to noise ratio. And one unoptimized detection can do this detection at 1 femtomolar. If you do fair comparison, use the similar techniques. If you change this particle to fluorescent dyes, we only can do 1 picomolar. So it's 1,000 times better than typical technology. We have really high sensitivity. So next, I will show you how can we get higher multiplexing. So one can use one dye labels for this different structures. Oh, excuse me. So when you change the different dyes, you will get different patterns. Then you can use the different Raman spectrum to label different probe, get different information about target. So one also can use the 2 dye labels. Then by varying the ratio of the 2 dyes, you can generate a bunch of new labels. And people will say, oh, you can do this for fluorescence dye. So I do the fair comparison. So I use exactly the same dye to do fluorescence. This is their spectrum. You can find, if you mix them, it's hard to get even more than 1. So this is the power of Raman scattering technology. So for the Raman thing, you can do three dye labels, and even more dye labels. Then we can do a math. How many labels are achievable by this technology? So assuming we have a 2 dye system, we have 5 energy levels. Theoretically, we can get 25, but you have to be careful about the portion which is not distinguishable with the other one. We can get 13th. Then how about the multi case? So although I couldn't write exactly formula to get this absolute number, but I can use this formula to estimate the numbers. So when we have 14 dyes, the number is at least 3 billion markers. Exactly is the C-value of human genome. That means if give me 14 well engineered Raman dyes, I can base label how human genome. It's an unbelievable number. So next, I will show you a demo to check the stability and suitability of this target, this detected format, which I chose 8 separate targets. So here's hepatitis A, hepatitis B, HIV, and the Ebola virus. And I use the CY3, TMR, and the Texas rat, and CY3.5. And here we have smallpox, we have anthrax, we have tularemia, we have cholera. So for this tool, we use 2 dye labels. So now we design 8 tests to carry out the stability of and selectivity of this detection format. So for the first test, all the targets are present. So we should get 8 spots. And in the second one, we take off this tool. And in third, we take off this tool. Now, how about our experiment result? So exactly, we found exactly follow this cartoon. However, if you see from this the black spot, you couldn't get which target is which. Then you do the Raman spectrum. You will find the difference between each target to the target, because you have on the probe you have different Raman labels. So this color is no meaning until you get off their real Raman spectrum. So by carefully check each of the spectrum, we found in this chip-based detection we do not have cross-hybridization, which information cannot be provided by this black and white 2-color systems. And this color is called we call, actually not we, it's a guy in analytical chemistry called Technicolor, but it's not Technicolor in Dream, that one from the movie. But this is a nice. So up to now, I talked about this things in one spot has one target. So next I will show you on one spot has two targets. Then we do the ratioing. So this spot has a two RNA target. By change the ratio, we can get this two peak intensity change, which is nearly it's a near relationship, nearly linear relationship between this ratio of two target and the intensity of the two intensities. A beauty of this Raman scattering method is you can use only one laser, because one laser is enough. And for the fluorescent dye case, you have to use multiple laser and to do multiple scan. It's really complicated. So here's one laser, one single scan. So up to now, so I show you this nanoparticle probes has a very high sensitivity, selectivity, and very high multiple labeling capabilities and ratioing capabilities. So I want to close my old chapter. Work is by this slide. So I have shown you that nanoparticles are such fascinating materials. They are important both in fundamental research and application, bioanalytical applications. And finally, I want to close my old research chapter by thanking these guys. First, my supervisor from Northwestern University, Professor Charles Merkin, and Professor Uli Bani at Hebrew University, and Professor Odette Milone and his student provide very important support for scanning tunneling microscope, and Professor Schatz provide theoretical support for our work, and that finger Professor Zheng provide very important discussion. And here is the brilliant student who was work with me, and I still have time, or how much?
5-10 minutes.
Okay, good. So this is my old chapter. So now it's a new chapter. My future work is still focused on the nanoparticles, but I have to change the directions. I want to do new things. And I hope some student here will join my group if I can get a position here. And first is about nanoparticle growth. Basically, I focus on the mechanism of this growth. And the second one is about nanoparticle assembly. The first is bioengineering applications. Specifically, is the first one is here. And this project, I think, should be interesting to you guys here. And in addition, it will be interesting to the students from chemistry department and biochemistry and material science. And I pursuing funding support from NSF and NIH and the DOD monies. And today, I think I do not have enough time, but I probably can say something about the third ones. So the first thing about this project, I light up the scientific questions. How can we make the nanoprobes with a bunch of different functions? And I use a simple approach. I call this a shell-lab approach. It's combinatorial things between different nanoparticles. I set off two short-term goals. The first is to make in vivo markers. Second is in vitro markers. So what they are? This multifunctional nanocomposite. They basically are polymer blocks. So inside the box has a bunch of nanoparticles with different functions. Say it has magnetic particles, it has fluorescence emitting particles, you have X-ray contrast agent particles. So that allow you has different functions. On the surface of this polymer box, we have the bioprobe for DNA, RNA, and protein detection. And the question is, why such research are important? So they can solve the current problem for using biomarkers to do detection. I already taught you about nanoparticle has bunch of advantage, but they also has some disadvantage. The first is slow hybridization kinetics. Compared with the fluorescent dye, it's really slow. Fluorescent dye can do at half an hour. We have to use few hours. Say it has at least 2 or 3 hours. And for the non-specific bonding, for our case, the gold particle case, we have very low. But for the using quantum dot case, the surface knot will pass through it. It's really high, non-specific bonding. So lower your detection limit, make your detection really high because you do not have contrast due to this non-specific bonding. And another bad thing for these gold particles, they have to rely on the silver staining, weird stuff. Once you buy a gene chip now, it's $120 for one, but if you silver staining one time, it's gone. So how can we use this nanoparticle probe to solve this problems? So the magnetic particle has another advantage. So it can tune this kinetic process by applying a magnetic field. So I use this salad, I call the shallot approach, combine these things together. So in this polymer box has the magnetic particles, also has fluorescent emitting particles. So for the fluorescent emitting particles, so you can use that as a optic tag to easily to detect the presence of this target. For the magnetic particles, that can provide you additional modulate of this probes. Once you play a magnetic field, so you can drop these probes on the surface of the chip. So basically, you increase the local concentration of the markers. So you increase the concentration of markers, basically you increase the kinetics process of this hybridization. Of course, you have the non-specific bonding. Then beautiful is you can play another direction of magnetic field. You can selectively to draw away, which is non-specific ones. So back and forth, by many times you can achieve increase the hybridization rate and eliminate non-specific bonding. Another beauty of this technique is you do not need to wash. Usually, people have to use stringency wash to clean this non-specific bonding. But if you use this magnetic field, you do not use stringency wash. Then usually stringency wash has a huge kinetic problem to lower your, lower your detection limit because you take off some real, which is a complementary part from this detection. Then, has time or not?
Just a couple minutes.
Okay. So now probably I just skip the things and how to make them, and basically it has a two-part. And first make this box with the particles, and then surface engineering of the particles. And okay, so this is the other strategy. And although this is a technologically research, but still we need concern about fundamental research, which is how can we make this nanocomposite with a uniform size and shape, and also with a uniform number of particles. So this is really important. Then this is surface engineering. It's another huge issue for this detection. So say if it has a very bad non-safe bonding, so you couldn't do successful detection. So first I will try this polymer strategy, which is increase the surface coverage of oligonucleotide, and which is really successful DNA detection in our method will be very useful for these things. And I won't go through this part. And finally, I will go through. I skip this part, and I will set some priority of research. So once I have one student, I will carry off this research. Then I have more students. I will do the nanoparticle assembly research, and then I have more money and will do these things. then summarize when all this proposal do good things, like what I imagine. So what I can get. The first is fundamentally understanding of nanoparticles, nucleation and growth, and a new approach for nanoparticle assembly, and the more efficient in vitro and in vivo markers. Thank you.
It's gonna be just